Hello.
Hi, Brad.
Hi, how are you?
Okay, we're on the,
Wait, what is it?
today's a holiday. I sent you 3 notes.
Oh, it's fine.
Oh,
It's Victoria Day. I totally forgot.
Victoria Day, yes, you're in Indiana.
yeah.
I mean, when I was in Canada, yeah, we would have this as a holiday. Yeah.
We celebrate Queen Victoria.
Yeah.
I mean, for us, we have Memorial Day, which was last week. No, it's next week. Memorial Day is
next week. Yeah. Right. Looks like we've got Dick and Hussain, Jesse, Vershali. Hello, Vershali. And Pakhi. All right.
Okay, Bradley, take a look at your email. There are 2 other people I invited, but only half an hour before.
Okay. All right. Yeah, I'll take a look.
Okay. They might show up. They might show up next week.
Okay, yeah, that would be fine. Yeah. So, any updates before we get into? There's Morgan.
Yeah, one of my notes from the stuff I did this weekend is you may be off the hook on Perlin noise.
Oh, okay. Well, I was going to give a demo on that, but okay. Well, I mean, it'd probably be useful for other things, but
maybe I need some independent thought about it.
Yeah.
But
yeah.
I think I found a way around it.
Okay. Yeah. Well, I'll show you what I have. We'll see if it's useful or not.
Okay.
I don't know if Hussain had anything he wanted to.
Yeah, I mean, so I've already presented a few times. I think everyone in this room, and obviously Dick, because it's under the project I'm working under for the breast cancer CT computer tomography research, accelerating that sort of work. So I mean, it's not about DevoWorm, but it's related to the.
It's okay.
So it's not.
May have roped Bradley into it too.
Yeah.
If it involves Perlin noise, then on some level it involves Perlin noise. So I mean, I've already spoken about it a couple of times, but I'm happy to just reiterate too. So overall, this project I've been working on there, and maybe I can share my screen so I can share like a paper too.
Yeah, go ahead.
Because that'd be pretty helpful.
All right.
I just need to pull up the paper. We'll just get that.
Okay.
Yeah. So excuse me. Breast cancer. Here we go. And this is from my inbox. So showing a paper from Gmail. Okay. So Dick and I have been working for the past couple months now on a very recent new project involving improving the efficiency of methods of breast cancer computer tomography. Excuse me. So with breast cancer research, there are a lot of different approaches to detecting or imaging or trying to see how well we can diagnose and detect breast cancer, like mammography, ultrasound, MRI, and like CT scans. So this approach we're investigating is using algorithms like MART, and potentially like maybe even like some other ones like eHector and advanced iterative methods for image reconstruction for detailed and high quality images from the CT scans themselves. So we want to overcome typical limitations of noise, of artifact. So the MART algorithm for breast cancer, in particular, like what was it, like multiplicative algebraic reconstruction technique, similar to like from this paper that I'm showing right here. Like this one I'm showing because Jake sent this one to me recently, but I wanted to give an overview of what a typical, say, machine learning or breast cancer post process. This isn't exactly what we're doing. This is from this paper, but ideally we'd want to investigate these methods. We want to investigate these methods. We'd want to investigate our approach to, if possible, see where we can go to town with this, and if we can patent this. create a startup, particularly using Janus spheres, which is really cool. And not only is it like really cool in and of itself, it's going to be used with 3D modeling tools. And speaking of which, Dick, I mentioned Blender. That cool art software program I told you. I'm trying to switch over to Unreal. I think Unreal would actually be better than this. So if you're familiar, Unreal's another graphics engine. Like it's used like in video games, the VFX, things like that. But like basically, we need to use some sort of three-dimensional engine for simulating X-ray beams to interact with the mirrors between the two hemispheres of the Janus and the Janus spheres. The Janus spheres are not on this. They're not on this one, but the Janus spheres themselves are basically excuse me. When we study how the electronic beams hit the mirrors between the two Janus spheres, that is what's involved in the breast cancer detection themselves. Like we would want to see how okay, so if I'm getting this right, it's the spheres themselves that are used in detecting which tissue would be considered cancerous and which one would not. So
not quite. The purpose of the Janus spheres is to aim the X-rays. We have detectors at the other end. They have to go through the breast.
Yeah. All right. Let me just take note of that. By the way. Yeah.
So what we're trying to do is electronically steer X-rays, which is quite a trick.
We're not pulling tricks here. This is all legitimate research.
Yeah.
But yes, it's fine. All right. Well,
Don't be so sure. We ain't peer reviewed yet.
Yeah.
This type of sleight of hand you can see in statistics and science, so I'm pretty shocking. So, well. Anyways, so okay. So speaking of tricks, so basically the Janus spheres, excuse me, after the Roman god Janus. So, and he was like depicted with two faces. So that's where you get this idea of like two hemispheres. By these nanoparticles, these spheres would be characterized by their dual functionality of two hemispheres, each from different materials or different types of surface properties. And you can use them. You can maybe use for multiple roles within like a single application, like drug delivery, image processing. So okay, so using the Janus spheres to steer the X-rays themselves. So typically we'd be able to model them with one side of a particular material for reflecting or deflecting X-rays, and the other one for like something for more control, like magnetic responsiveness. Let me actually see, so okay, and this is not relevant, but this is just another approach. So this one that Dick sent me is like the so we would see patenting or intellectual property. We would want to see compared to others. Yeah, I'm happy to answer questions. Happy to see what sort of discussion or things people might be interested in talking about moving forward. So yeah, in Blender you can easily make spheres and things using like the 3D art and animation software, and but Unreal, I found Unreal is actually better for physics simulations. Okay.
Oh yeah.
So, like these companies for like software engineering, physics, engineering that have been potentially reaching out for interviews and potential positions, many of them are really strongly preferring Unreal. It just has a lot better functionality for physics simulations compared to Blender. So Blender's decent too. But I think Unreal just offers a lot more. So that's all.
Yeah.
Based in C++. So Blender is primarily based in Python. Yep.
Yeah.
Excuse me. Yeah.
That's great. Yeah. Any other comments on that? I know that this is something that Dick has been trying to figure out what methods to use to best do this. So.
Yeah, for sure. And this actually is a pretty strong intersection of some of the work that other people around here are doing too. For example, Bradley's work on multi-scale Perlin noise, based off, say, we want to see how we can integrate Perlin noise based on like the anatomy simulations that are already there for enhancing the realism of tissue simulation, and see which ones can provide like the best contrast or interaction with X-ray protons too. Excuse me, the X-ray photons. So yeah.
Hussain, this weekend I went through a whole bunch of papers on contrast enhancement, and it looks like we may be able to ignore Perlin noise.
All right, cool.
Okay, I'll send you a copy of that later today.
Cool. Thanks.
Okay, I already sent a copy to Bradley because he was working on Perlin noise.
Yeah. Yeah. Okay. Yeah, that's great. Thank you, Hussain. Yeah. So let's see. Let me get into a few things before I get into I was going to show the stuff I've been doing on Perlin noise. I have a demo, but I'm going to show a few things before that. So this week is, I guess, our final week before the end of the community period in Google Summer of Code. So we have Google Summer of Code, we have our community period. It's about 3 weeks where, we try to get familiar with our community. And so it's a way to kind of figure out, set up what you're doing, but also to make connections with people in the broader community. In our community, we have DevoWorm, and we have the DevoWorm group, and we have our website and our YouTube channel and our GitHub repositories and all that. We have our meetings. And then we're also affiliated with the OpenWorm Foundation. So the OpenWorm Foundation is, what we're organized under. And Pakhi and Mehul have been attending our open source meetings through the Orthogonal Lab, which is another thing that we're affiliated with. But today I wanted to talk a little bit about OpenWorm to make sure that we, as a group, are sort of on top of that in terms of the community. So I'm just going to go over the OpenWorm website, which is, I guess, somewhat out of date. It could always be updated. We kind of rely on people to make pull requests to the website to update it. But, they're different. So they're different. First of all, OpenWorm is organized into a number of different projects. It's a foundation. It's a 501c3, so they have, that sort of structure. It's just a collective of different projects. So, we have various things available to people, like the OpenWorm browser, which is modeling C. elegans in 3D. So you can build 3D models of the worm. You can build models of the muscles, and you can build models of the neurons and the cells and the body wall. And that's a static model that you can rotate and cut apart and look inside. But we also have other types of models that model the physiology and the cell biology more specifically. So we have the OpenWorm browser, which is a nice intro to the worm. And that's something that's been maintained in Blender. There's a master Blender file that has been maintained for, the duration of the OpenWorm endeavor. So we have other models as well. So we have the NeuroML Connectome, and this is a model of the connectome, or all the neurons of C. elegans. Yeah. So this is 302 neurons with their connections here, and you have, the idea is to model each one of those, each set of connections, and you're able to explore the connectome in different ways. So one of the reasons that C. elegans is interesting is because we have this relatively small connectome with the, we have a tractable number of neurons, we have a tractable number of connections, and then we can actually model that in different, behaviors, like movement behaviors or whatever. So we have, these kind of tools like connectome modeling. We have visualization and simulation platforms like Geppetto. That's another thing that's being worked on. This allows you to do different types of simulations in NeuroML, and so you can play with those. Let's see if we have any other projects off of this. Okay, Cybernetic, which is the biophysics model of the worm. So this is where we have a simulated worm, and instead of having a 3D model of different parts of the anatomy, we have this worm that has a physics, and it's moving through a physical medium. So it could be moving through a gel, or it could be moving through soil. And the idea of cybernetic is you model all the physical parameters of those interactions between the worm's body and the surface. And so, you have these kind of biophysics models. You can also model parts of the muscle, parts of the soft tissue. The soft tissues are these compliant, it's a compliant material that has a variable stiffness. And depending on the medium, there can be some interactions there that are interesting and affect movement. So you generate movement from the connectome, but there's also feedback from this physical environment. And so we can model those things. There's worm sim, so that's another type of mechanical model, and it's focused on muscle models and things like that. So we can take these muscles that we saw in the 3D model, and we can model their dynamics. We can model the Hodgkin-Huxley dynamics of muscle and so forth. So we can do a lot of things here in the we also use smooth particle hydrodynamics to model some of these things as well. Then there's, of course, DevoWorm. And, so there are a number of projects, and you can go to the website at openworm.org. You can explore some of these things. We have different databases, like the movement database. We have different data sets that I talked about last week. And, of course, if you go into the Slack, explore the channels and see what's going on. There isn't a huge amount of activity, but because, we have the paid version of Slack for this, we can go back into the archives and look at some of the things that people have posted on some of these projects. And if you go to the GitHub repository, you'll see that there's a fair amount of activity. So this is, there's a link directly to the GitHub repository here. And this is, of course, where you can do things. You can contribute to OpenWorm proper, because a lot of the GitHub infrastructure I've shown in the meeting is DevoWorm related, but this is the OpenWorm GitHub, and so I don't know how many people are really familiar with what's here, but there's a lot of the projects are here. C302, which is one of the connectome models. Geppetto is here. Channel Worm, which involves ion channels and modeling ion channels here. We have other repositories for different papers. So there are some standard biophysical models in the literature that are relevant to C. elegans. And what's been done is that we have these repositories that are created. These model DB repositories, and you can look at those and maybe even contribute or make a fork of your own. We have this main OpenWorm repository, which is where you can find a Docker file. And that Docker file, if you install it on your local machine, allows you to run a simulation that kind of goes through all of the models. So it's a summary of the models, and it's kind of it's a tough thing to get running, but if you run it, it's really nice demonstration of what the foundation is all about. Then there's some repositories for some of the side projects. There's nothing here for DevoWorm because we're kind of separate from this GitHub repository. And so, there are all sorts of resources here. There's a fork of the Worm NeuroAtlas and some other things. So that's a lot of the stuff that's involved in the community of OpenWorm. There's also, of course, the Slack and, of course, the website, which has some links to documentation. And like I said, it's a little dated, but, it's something that has, I think, most of the relevant information. We have like papers and there's a science link that links to, a list of papers and some documentation. So that's the OpenWorm Foundation. Yeah, so we had a DevoWorm about a decade ago, but we've been kind of going separately from the main OpenWorm apparatus. Although we're still part of what they do. So yeah, so definitely check that out if you're interested in that community. So yeah, community period will be wrapping up probably at the end of this week, beginning of next week. Next week, of course, is the in the United States, it's Memorial Day, so we may or may not have a meeting. It's hard to say. But then we'll get into the coding period. At the end, towards the end of May. So actually, for Pakhi and Mahul, I expect you to be working kind of right now on getting set up, getting everything ready, and I think you have been doing that. And kind of working through some of the things that you're going to be doing for your coding period. And I know Himanshu's here and Jess are here. So as mentors, we want to be able to help people. I don't know if you need to help them with the coding part, but we're going to be facilitating our meetings, and we'll have the open source meetings on Fridays, and we'll have these meetings on Mondays. So try to attend at least one of those, but preferably both, because I think that the open source meetings are valuable, and then these meetings are valuable for kind of understanding what's going on in the project. And we'll give updates, and we'll see. We'll do this weekly, and we'll see what kind of problems arise, and we'll see what kind of progress is made, and then we'll kind of constantly reevaluate over the summer where we are. So we want to really keep on top of that, so that we don't have any major problems that arise, because sometimes that can happen if you fall out of communication or you just kind of ignore small problems. So we don't want to have that happen. Also, for Pakhi and Mahul, we have this is the place where I want you to contribute. I've invited both of you via email to DevoGraph or to the DevoLearn organization. So DevoLearn is this organization, github.com/DevoLearn, and it has the DevoLearn main repository, some of the cell models, like we have CellSam, Segment Anything, we have the Nucleus Segmenter, we have Mono Annotation, we have a number of other models here. So basically, these models, Nuclear Segmenter, Membrane Segmenter, and Lineage Population, these are the same things that you'd find on Hugging Face. The Cell Segment Anything model is something that Sushmant was working on last year. That's a fork of the Segment Anything model from Meta. And then we have some other things. And then that's all kind of the DevoLearn project, or the DevoLearn platform. DevoGraph, of course, is the repository you'll be contributing to, and that's here. So we have a number of contributors that have already been a part of this, but I've invited you to DevoLearn as the organization. That'll give you permissions to DevoGraph. If you accept that that invitation, you'll be able to push to DevoGraph and manage issues, which we will manage on a project board, which I don't know if I've set it up on this one. I may have to go to the main. Okay, so I don't think I've set it up yet, but I'm going to set up a project board in DevoGraph where it's actually managed at the organizational level. But we'll have that project board ready to go, and it'll just be something we'll review and see. If you have issues, please post those issues, and then we can put them in a Kanban board so we can move them across from to do to completion. And that'd be a good way to manage some of these issues, especially the ones that are collaborative, where we're all kind of working on different things that kind of overlap. So if there's a common resource that's needed, or there's something that might be of interest to someone else, that would be a good place to put it on the project board. And so, for the repository, I kind of want people to push regularly to that, because it's important that we keep everything off of our local machines. And the reason for that is that, you don't want it crashing and losing all your work. You want to have everything kind of pushed to GitHub, pushed to an open source place where A, we can evaluate it, and B, it's not, it's backed up, and we don't lose anything. So that, and also it kind of reinforces workflows because, what's important here isn't just that you generate a bunch of code, it's that you kind of sculpt a workflow so that we can later write documentation for it. We can go back to it and see what the logic of something is, how something was arrived at, and what everything, what all is there. So it's very important. Yeah. So any questions about that? Yeah. All right. So yeah. So let's see. Next, I'm going to talk about something that. So my knock Deb was one of our GSoC students in a couple years ago, and he really worked on driving forward the DevoWorm stuff. And he's, him and Minock or him and Mayok are brothers, and they kind of came in one year after the other, so they're both very key to developing DevoWorm as it stands now. Well, they keep in touch in the organization, so after GSoC they keep in touch, and Minock just sent me this paper and this associated PyP repository on CellSeg 3D. So this is on self-supervised 3D cell segmentation for microscopy. So this is a new thing that came out recently, I guess. This is something that is, there's a platform called Napari, or a set of tools called Napari. And this is, it's a Python package that plugs into Napari. So this is CellSeg 3D. So a lot of times when people are working with microscopy data, they need to have a number of tools. But one of the key tools for this is cell segmentation. And so as doing your DevoGraph work, step one will be kind of working with what we have in terms of cell segmentation and applying that to your problem. So you have to have a good starting point with input data, with getting that input data, getting the features out of it, and then moving forward. And when we did DevoGraph projects in the past, we always had people working on the cell segmentation as part of their project. But this year, I think we have enough infrastructure locally in DevoWorm to sort of skip that step and just apply what's there. So we'll see how that goes. But in any case, this is a new package called CellSeg 3D. And this is from this group here, group of authors. And let's go over the abstract and then go look at the PyP repository. So the abstract for this is understanding the complex three-dimensional structure of cells is crucial across many disciplines of biology and especially neuroscience. So this is focused on neurons and segmenting neuronal cells. And of course, those are very different from developmental cells in the sense that they have different morphologies and they're, embedded in a different type of phenotype or different. I mean, you're probably you're doing mammalian nervous systems or, those are much more complex than something like what we'd see with C. elegans. Here we introduce a novel 3D self-supervised learning method designed to address the inherent complexity of quantifying cells in 3D volumes. So again, we have these three-dimensional volumes. We have these two-dimensional images that are in a stack, usually. So you have a stack from one along one morphological or phenotype or anatomical orientation, and then you go from maybe from top to bottom, and you get these. You get different parts of the tissue in those snapshots, and then you reconstruct the volume from that. So there's a lot of complexity in these 3D volumes, and what they'll do in preparing the specimen is they'll clear the tissue, try to get a lot of the extraneous proteins and extracellular matrix out, so you get a clear snapshot of the cells. And so then they do sectioning, and then they take these images, and you get this data. So we offer a new 3D mesospim data set. So this is a technique called mesospim. SPIM is a type of microscopy that yields pretty high resolution images that usually don't have, are not associated with a fluorescent signal. It's just like a image, usually like black and white, and it's very high resolution. We have some SPIM images of C. elegans in our DevoZoo. So, but this is a technique that you can use to really kind of get a handle on the cells and the cell bodies. And so we offer a new mesospim data set. And so they actually have a data set associated with this as well, and show that CellSeg 3D can match state-of-the-art supervised methods. So this is something that where, there are these state-of-the-art methods, and you want to be able to match them, and this platform does that. Our contributions are made accessible through a Python package, which we'll talk about in a minute, with full GUI or graphical user interface integration in Napari. So if you're using Napari, the platform, this is basically a plugin. Yeah, so there's a data set associated with this. This is just going to this preprint finding. I guess it's in their supplemental materials. So this is the PyP repository for CellSeg 3D. This is associated with Napari. So this kind of gives you an idea of what kinds of things that they're doing here. So this is the logo that they have here. And then this is, if I can zoom in on this, this is sort of an image of what you see. So you're working with this three-dimensional volume, and there are these different volumes here, and they all have, cells segmented in those volumes, and then you're able to work with them. So you install this. There's documentation here. This is from the Adaptive Motor Control Lab, and this kind of deals, this kind of talks about the details of the software. So this looks like an interesting approach. Let me look at the documentation. Maybe that has the data set in it. Okay, so now you can see up here this 3D volume. You can zoom in. This is a human brain. You're able to take a volume of the human brain. You're able to take a volume of that human brain and focus in on it. So you have an atlas that you can match up things to. So that's good stuff. I don't know, I don't know how relevant that is to what we're doing this summer, but these are the kinds of tools that you can use. That this is sort of the state of the art. So I think being familiar with the state of the art in these areas is good, because kind of what people are trying to do or want to do, and then incorporate those features. So you can see that like this exercise of cell segmentation is not just as simple as throwing data at an algorithm. There's a lot of stuff you need to think about in terms of the data set you select, and then also the sort of the techniques that go into pre-processing, as well as linking the segmented data to some atlas or some other thing. So, and then of course in our case, we're taking segmented data and then we're doing other things with it. And so that's another workflow you need to plug into. Okay, so that's enough on the stuff from Mynock. Thank you, Mynock, for sharing that. Now I'll get into this Perlin noise demo, because we've been talking about this for a while, and I've been having some problems finding the right code for this. There are some toolboxes, and there's a lot of deprecation that happened with some of the toolboxes available. There's a toolbox in Python called Perlin noise. So you can install Perlin noise and work with it. And, this is a toolbox that's freely available. So what I have here now is a Colab notebook, and I've installed Perlin noise. I've got matplotlib. I'm going to be generating some plots. And then, so that's all set up. Perlin noise 1.12. So the first thing I'm going to do is I'm going to run this Perlin noise function, and I'm going to, there are two variables here. There's octaves and seed. So octaves is in Perlin noise. So what Perlin noise is, it's this specific type of noise that was generated by a computer scientist named Ken Perlin. And he developed this as a procedural generation tool. So you have what you know as Gaussian noise, which is, if you've ever seen television static, that's what that is. It's like you can have an even field, and we'll see that in a bit. But that's what Gaussian noise is. It's basically this uniform noise, and it's across. You can have a two-dimensional plot of it. Perlin noise is different. Perlin noise has sort of a periodicity to it, and it has this pattern. So you can see here in this plot in green, you have this sort of, it looks like maybe it's a pattern, but it's sort of blurry. And so it's generating this sort of, it doesn't look like static. It looks like it has maybe even some pattern underneath. But it's, maybe it looks like blobs. If you've ever seen bubble tea, where they have these balls in the bottom, and that's what it kind of looks like. But so it has these different blobs, and then if you increase the number of octaves, you actually get something that looks more like, it becomes, as you increase the number of octaves, it looks increasingly more like Gaussian noise. So this is one of the parameters, octaves. So you can set your octaves high or low, and it gives you different sort of, patterns in this noise. And then the random seed, of course, is just a number that you plug in. And I don't know if there are any tendencies in the seed. Usually not. It's usually a random seed. But you want to, if you vary that, it varies the pattern in some way. So what I'm going to do here is generate a 100 by 100 pixel array. I'm going to generate Perlin noise using this toolbox, using this function Perlin noise. I'm going to do this for an octaves value of 4. And then I'm going to generate this pick variable. I'm going to plug that into this IM show. I'm going to color it green, and there you are. That's the first image. So you have this Perlin noise that's in green. You have what looks like these dark blobs underneath. You have these lighter areas, and it's very blurry. And the idea is that you have these octaves, which are these, this periodicity within the field of noise. And so it's very different from that uniform static. And so that's what it looks like using this Python tool. Now we have this, so basically I'm going to do some checks here. The noise is at a value of 0.5, so this is basically, I could probably bias, if I wanted to go into the function, I could bias the noise in some way. I could also do some sort of, it's not limited in terms of spatial dimension. It's seamless for any size, so if I increase this array by, and I'm going to do this later, by 5-fold, it's going to give me a similar pattern, So it's not going to repeat. It's just going to kind of be the same pattern, but it's going to be bigger. So that's basically what's going to happen here. So this next step is where I do a Perlin noise plot with a higher octave value, which is 25. So this is actually, almost a little bit more than 5-fold greater octaves. So this is going to show how if you increase the octaves parameter, it looks different than the first image. It doesn't actually necessarily look like uniform static, but it looks actually more uniform here. You'll see this plot in green. So again, I'm plotting 100 by 100 pixels, and we have this image in green where you have, it almost looks like a shag carpet, where it's got like this pattern. It's not, it's highly periodic, but it's not, it's uniform, I guess. It's periodic in some way, but you can still see it looks different than like a static Gaussian noise. So this is interesting. This is just generating this. It's just procedural generation, and you're just changing one parameter here. course, I'm changing the seed, but I don't think that matters so much. So this is what we get here. So now I can also do this for a one-dimensional array. I can plot this in a one-dimensional stretch. So, this is 100 by 100. You can plot this for a single one-dimensional array of Perlin noise. And now why this might be relevant is that you can also generate one-dimensional cellular automata. And I was thinking about this when I was doing these experiments. I was thinking about cellular automata. And like I was thinking about this one-dimensional case of cellular automata where you have this like long, basically it's a single vector. And you have this one-dimensional cellular automata where you have two neighbors, a neighborhood of two cells on either side of you, and you can get some interesting patterns, and then you put those together, and you can get the kinds of things we see from the Wolfram books and things like that. So I was thinking about this because in cellular automata you have certain rules, and you have these cells that change their state over time. And with Perlin noise, it's generative as well, where you have this procedural noise that you can, get a sort of a end state. And so what I'm thinking is that there might be some connection between Perlin noise and one-dimensional cellular automata. But that's something for a different time. I don't have a demo for that. In any case, so this is the code for the one-dimensional Perlin noise array. This is actually where we have our random array. So this is actually random noise here. This is not Perlin noise, and this is now in blue. So this is where I'm generating a random array of 100 by 100 pixels, and I'm plotting it in blue. And you can see that it looks like this uniform static. This is what, if you watch an analog television and the station is off the air, this is what it looks like. It has a static field. And, it's just kind of, there's no periodicity here, no pattern, but it's actually useful as a mask. So you can mask things, and people often have done this, where they mask it with this uniform noise. And, an image, you can embed an image underneath that noise, and you can increase the noise, at a certain, and we're gonna see that later. And so, this is a very useful tool, but we can also use the Perlin noise as a mask, and we're going to see how those two things vary in a little bit. So that's random noise, and then this is a comparison of random noise and Perlin noise back to back. So this is where we have this random noise but in green, and then we have the Perlin noise underneath. And so, these are two different types of noise side by side. So you can see the differences in how they look. This almost looks like an artificial turf type thing, and this looks very different. This almost looks like maybe a set of bacterial colonies if you squint closely enough. But the point is that they're different. So we have this, now I'm plotting things in red here. This is our random, but what we're doing here is we're doing some experiments in combining Perlin noise and Gaussian noise. So, we can see that there are differences here, but what happens if we combine the two? What happens if we overlay the two instead of putting them side by side? And so what we see here is the first example is where we have this additive combination of Perlin noise and Gaussian noise. And so we have this, I've just done an additive version here where we have additive noise, and it looks different than the Perlin noise. It's not, it doesn't look like either one, but it looks maybe closer to Perlin noise. So this is what it looks like here. And then the second example is where I have a multiplicative example. So it's just the difference between taking these two arrays and adding their values together, and taking these two arrays and multiplying the values together. And so the multiplicative case actually looks quite a bit different than the additive case. And you can see in the multiplicative case, you almost get this like enhanced Perlin noise. It almost looks like it's just kind of like got a weird mask on it. And so you have these sort of, you can see kind of the high octave Perlin noise with sort of this mask of noise, Gaussian noise over it. Anyways, it looks interesting. So that's interesting from the standpoint of different types of noise and what you might be able to do with Perlin noise. But what I want to do now is I want to look at like what happens if you mask an image into that, or you mask it with Perlin noise, you mask it with grading of bars, and you mask it with Gaussian noise. So I'm going to use Mario here as an example of an image. It's very colorful, has a white background, and we're going to take Mario and we're going to mask him with different types of noise. So here's Mario, and we have our Perlin noise. We have octave value of 72, so it's very high octave Perlin noise. And we're generating the Perlin noise here in green. You can see that this is even higher in terms of the octaves than even the high octave example that we had in the last set of examples. So this is where you actually see some, almost like some striping underneath. You're starting to get some real, periodicity here and some consequences of that. So this is the mask that we're going to use. And so we take this mask and we take Mario and we place them over one another. Now the way to do this is you can actually do this using plot.im_show from Matplotlib. So you can use Matplotlib as the tool. You can actually take Mario as the underlying image. You can then use the noise as a mask. And you can actually use it in different colors. So you can do the CMap function. You can use like different colors to do this. So you can play around with this. But the point is this alpha parameter. So this alpha parameter is sort of the transparency, or it's the intensity of the noise mask. So this is a high alpha mask. So this is where it's almost 1. So 1 is like the maximum amount of noise. 0.0 is the minimum amount of noise. Or I guess you shut it off at that point. But you can see that Mario is pretty well masked, but you can still see the underlying pattern. So this is this high octave Perlin noise masking Mario, and you can see Mario, but you can't see, you can't see the detail. You can only see the outline. Now we're going to take random noise. So we're going to take a random mask in green, and you can see it looks very staticky and uniform. And we're going to mask Mario with that. So now you can see Mario again with the same alpha value, but you can see that it looks, it's more detectable than you can see with the Perlin noise. So the Perlin noise is actually obscuring the underlying image more than in this case. Because in this case, at least to my eye, you can see like the details of Mario's outfit and so forth. So this is, just different types of noise and how they behave when you're trying to mask them at the same alpha value. So those are those examples. And then now I'm going to take some Perlin noise, some very low octave Perlin noise, an octave value of 4, and I'm going to go back to that. So this is the first example I showed, but I'm going to replot that. And notice that, we have this, it's a different type of pattern almost. It has these blobs, and it's very blurry looking. And so then we're going to take Mario, we're going to mask Mario with this noise at the same alpha value. And we now have Mario masked. And it looks weird because we have this jet map instead of the green, but it's just to accentuate sort of the mask. And so we can see that there are these blobs that are different colors. So actually at this frequency, it doesn't mask all that much of Mario, except that the blobs actually mask quite a bit. So it's very, it's non-uniform in terms of how it's masking Mario's image. So that's very interesting stuff. Then we have this, now I want to get into this grading. So, a lot of times in perceptual studies, they'll use these bar gradings, and they'll have people like look at them and see if they can find patterns behind them, or sometimes they'll try to detect the bars. So what I've done here is I've created a bar plot where it's, very, very dense. So each one, for each value on the x-axis, there's a bar. And so there's so many bars here, you can't really make out the bars. If you really squint, you can kind of see there's these bars, but you can't really detect them. And that's actually what we want. We want this very dense packing of bars, this dense grading. And so what I'm going to do with this dense grading is use it as a mask, but this time against the Perlin noise, so I can see what will mask the Perlin noise. And so now I've used this low density, this low octave Perlin noise, and I mask it with this high density bar grading. And so you can see that the bar grading covers up the Perlin noise, not to the extent that I can't see the underlying pattern, but it kind of covers up sort of it tints the color map here. So, it's interesting how these things interact. So that's all I have. I'm just, I'm playing around with Perlin noise. I'm getting some interesting results. And, if we don't use it for imaging, we can use it for a lot of other things. I'd like to play around with that some more and see where that goes. So let's see. Okay. So I have some things here. Yeah. Dick said, this is a message for Hussain. DevoWorm has expanded to many other organisms. Yeah, so I think last week I talked about it, but especially in this 10 years of DevoWorm presentation that's on our YouTube channel, I have a slide on all the different organisms that we've worked on. So I've noticed in doing this inventory of our different organisms, we've done like 8 different organisms that we've kind of worked on data-wise. We talked about a lot more. And then we've also talked about digital organisms, which are kind of a 9th class of organisms. So we do a lot of comparative work. We talk about a lot of different types of organisms. Morgan says, I dropped Loic Royer's Napari Chat GPT plugin and YouTube presentation in OpenWorm Slack. So yeah, this is Loic Royer does some stuff with this Napari. And then the Chat GPT plugin, which of course is obligatory now. And then there's a YouTube presentation. So that's in the OpenWorm Slack. It's very interesting. I know Morgan, I don't know if he has anything to say about that, if he's familiar with Napari tools.
I saw his presentation at the Advanced Microscopy Workshop at Berkeley, and it was pretty impressive as kind of a real-time code generation, like on-the-fly image filtering, and it was pretty cool. Anyway, but yes, now it's standard that you have ChatGPT in something. So.
Yeah.
But he showed up, and I dropped a link to his Janelia code review of it. Like it was also just kind of interesting to see his process.
Yeah. Yeah, I think that'd be great. Even though it's not really like what we do here, I think there are a lot of interesting things with imaging and, just neuroimaging, but also like CT and other types of imaging as well.
Yeah, sure. Yeah, and that was an interesting presentation on that kind of noise. I wasn't familiar.
Yeah.
But it reminded me of various Gaussian random fields, smooth maps. And
those are really big. Those are really big in spatial computing right now.
Yeah.
Bradley, are you familiar with Ising lattices?
Yeah.
Okay. And a one-dimensional Ising lattice has an enormous number of models for it.
Yeah.
And if you look at that versus time, it will possibly generate patterns similar to in some way similar to what you were doing there with the noise.
Right.
Well, it might be worth looking into. As far as I can tell, nobody has ever done it. Even though I found 800 papers on one-dimensional Ising lattices.
Right.
Okay, so it might be fun doing. The different models for Ising lattices have all sorts of interactions between the points.
Yeah.
The Ising models. The Ising models. The Ising lattices and the models. I always used to find especially when I was studying like physics and like computational applications of physics in various areas, I found that there are problems related to the problem of degeneracy, like which stages are at which you can state that they're degenerate. I found that those were pretty damn interesting, from a 3D spatial perspective, like when involved in spatial computing. So those are pretty cool. Back to what Morgan was just talking about, like Gaussian spotting, and you were talking about that, right? Like Gaussian spotting, and that stuff is that's really big. That stuff is really big, not just from a research point of view, but from the current software technologies that are involved in 3D imaging. So like even like augmented reality applications. They're all absolutely discussing that when generating images, and being able to scan my face and create a deep fake of me. Yeah. So Microsoft just released one of their abilities to do that from a single image. Take someone's image and you can make that person say whatever the hell they want and create a video of it.
Yeah.
And like you can also like kind of deduce like a voice, or you can give it a sample voice too. But anyways, yeah, like Gaussian spotting in the application of image processing with the machine learning and neural networks that are involved in that capability. That's where it's all really picking up. The Ising models and Ising lattices too. Yeah, that's really cool too. So we'd like to definitely see where things go. So
Yeah.
The idea here is an Ising lattice changes over time. If you have a 1D Ising lattice, you can make it two-dimensional by making time the other dimension. And then you get a pattern. And these patterns may or may not be related to the Wolfram patterns.
Yeah.
Okay, so I think they're worth exploring.
Yeah. Yeah, I mean, yeah, like Wolfram has like these rules that he classified, and there might be rules that are kind of in these patterns, like because it's not it's not like I mean, I know it's like random. Well, it's procedurally generated, so it's sort of at random, but it's quasi-random in the sense that it has some structure to it, I guess. Yeah. So.
Yeah, and the Perlin noise has some structure to it too.
Right.
So it'll be interesting to take your 1D Perlin noise, and I guess the Perlin noise is kind of static. You generate a picture and that's it.
Yeah, well, it's procedurally generated, but it doesn't have like time steps.
Yeah. It doesn't have time steps, whereas the Ising lattices have time steps. And the time steps can be the Y coordinate.
Oh yeah.
And therefore generate a picture.
Yeah.
I've tried to bring that up at a remote Ising lattice meeting, and nobody seemed to understand it.
Oh well.
So
yeah,
I always say, unfortunately, if no one understands it, perhaps you could try explaining it differently.
yeah, I know, but
I'm trying to. Yeah.
You have limited time.
It's difficult. Oh, if you mean they can't understand it from their own lack of background, then that is different. But
Well, a two-dimensional. There's a big difference between a 1D and a two-dimensional Ising lattice. You know that from your physics. Of course. A standard Ising. Ising himself invented this as a one-dimensional model for ferromagnetism, and it didn't work. And then other people made it two-dimensional, and all of a sudden they were getting real phase transitions. Okay, and now there's a huge literature, even on 1D Ising lattices, and with different direction rules.
Yeah, I mean, and even then, excuse me, from a computing perspective, it's all pretty straightforward. Like as soon as you understand the math and you can show that it's verified from a theoretical point of view, you can just code it as you need to, like Python or Julia or R or something similar. So that's there. And I guess from a more theoretical perspective, it's like also in some ways understanding. I like to really know, like, is it how valid are the constructive proofs? Like, can you really show degeneracy? Can you really show the patterns themselves follow from those rules as established? And under what conditions, and how well do they really generalize to the general nature of Ising lattices themselves? By the way, real quickly, just wanted to show, this is what the Roman god Janus looks like.
Oh, okay. Yeah.
Like, so I was talking about student faces. And with two different chemical properties, one on both, one on the other. So
do not ordinarily give phase transitions, but if they had an infinite range for the interactions between particles, they would do that. You can get almost the equivalent of that by taking the one-dimensionalizing lattice and turning it into a circle. And then each particle can interact with itself.
Oh, yeah.
And other. Okay.
Yeah, I mean, you call it like either a self-referential interaction, like a it would be its own system, like that, the particle.
Yeah. Okay. Now, curiously, I used this as a model for the shapes of cells called archaea.
Okay.
And these are often in polygonal shapes. And I was able to show that you get polygonal shapes by doing that. So in other words, the circle transforms into a polygon.
Yeah.
And you can actually model this physically if you have a bunch of little magnets and put them in a ring.
Yeah.
And they will snap into polygons.
Yeah, yeah.
So it seems like a plausible model.
Yeah, like it's those transformations, and they're essential too. So yeah.
Yeah. So it's a lot of fun.
Yeah. Sorry.
Okay, Bradley, I have to
Yeah.
get to another meeting. It's called coffee.
Oh, yeah. All right. All right. Well, thank you for attending, and see everyone next week. Thank you.
Okay, thanks a lot. Bye.
All right, bye.
Thank you. Bye.
Thank you. Bye. All right. Now I'd like to talk about Swarmalators. And this is a physics tool that allows us to look at swarming behaviors in a multitude of different contexts, different types of organisms, different collective behavioral systems, and so forth. So we'll cover a couple of key papers, go revisit some of the things we've talked about in previous meetings. We've talked about Swarmalators in a number of different meetings in the past. The first is a feature that we did on chemical flows and swarms, and this was back in March 6th of 2023. So we talked about Jeffrey's microswimmers and flows. We talked about Swarmalators as a concept, and we talked about swimmer organisms that exhibit chemosensation. So the Swarmalators paper was this paper here, diverse behaviors in non-uniform chiral and non-chiral Swarmalators. The Jeffrey's microswimmers are these, this is where we use Jeffrey's equations to model the orientation dynamics of an asymmetric object flow at low Reynolds numbers. So these microswimmers are swimming in this very low Reynolds number regime. And so it's not a turbulent regime. And so we're modeling sort of the way that they manage those flows, their orientation dynamics, and so forth. Related to that then is chemosensation and chemotaxis. So a three-sphere swimmer that exhibits chemotaxis and chemoreception and therefore directional behavior. Our second example, this was from March 21, 2022, and this was in a feature called mechanical programming. So we had a number of papers in this collection, including this one here, which talked about a pattern-forming cortical circuit based on Rho, F-actin, ECT2, and RGA3/4. We also had this paper on excitable mechanics embodied in a walking cilium. We had this paper on ciliary flocking and emergent instabilities, enabling collective agility in non-neuromuscular animals. So this is, again, this collective behavior. It's not always about swarms. It's kind of about organisms at very small scales, orienting themselves, traveling in flows, and exhibiting these kind of taxic behaviors. And so this was from the Manu Prakash group at Stanford. I remember that we had a number of papers from that group. This is another one, mobile defects born from an energy cascade, shaping the locomotive behavior of a headless animal. So again, your organisms don't have to have a brain, but they have to have that chemical signaling ability, or they have to have some sort of light signaling ability, the ability to pick up these sort of gradients and so forth. And this one is the swimming rheometer, self-propulsion or freely suspended swimmer enabled by viscoelastic normal stresses. So this is again is behavior where physics dominates, and we get these. So this is where we have self-propulsion at low Reynolds numbers. It's notoriously restricted. This is commonly known as the scallop theorem. So people actually have a name for this. Here we present a truly self-propelled swimmer, force and torque free, that will unable to swim in a Newtonian fluid due to the scallop theorem. So we know that in Newtonian regimes, swimming in low Reynolds numbers, if you don't have these sort of abilities to generate force or torque, then you can't swim. It's kind of a problem for some of these microorganisms. These organisms that they're talking about here, these swimmers, are able to propel themselves in a non-Newtonian fluid as a result of fluid elasticity. So they're able to sort of, what they have here are robotic swimmers that exploit these properties of the fluid that they're in to swim. And so these robotic swimmers have a head sphere and a tail sphere. So they're two spheres with a polarity. It's kind of like a very small two-cell embryo. The swimming speed is shown to have reasonable agreement with a microhydrodynamic asymptotic theory and numerical simulations. So this is hardcore. This is hardcore hydrodynamics. So the third feature is a soft collective matter feature from March 7 of 2022. And again, we have two papers here. We actually have a paper on a nematode called Tubatrix seseti. So these are synchronized oscillations in these swarms. If you look at the cover of Soft Matter for that month, these are these nematodes, and they're kind of behaving in this collective way. And this other paper, collective motion of cells model as ring polymers. So, unlike nematodes, these are polymers. These are not, they don't have a brain, but they can be. Okay. In this paper, collective motion of cells modeled as ring polymers. So in this case, we have cells, and we can model them as these kind of shaped polymer structures. And so the first thing I'd like to do is go to the Wikipedia page for Swarmalators. So this is a large concept, or sort of a unifying concept called Swarmalators. What I've shown you are examples of the Swarmalator concept, and we'll talk about sort of a seed paper that was written by Steven Sturgatz and colleagues, and another paper on the topic as well. This is basically, they define Swarmalators as generalizations of phase oscillators that swarm around in space and synchronize in time. So this is where we have the swarming behaviors, collective behavior, that gets synchronized in time. So a classic example of this are fireflies. They synchronize their flashing over time. They may exist in sort of the same area, patch of your lawn, and they basically will light up their luciferase sac to signal other fireflies, and so they can synchronize those over time. And so we can use a number of computational models, physics models, to model this, and we'll see in a bit what those are. Karamoto oscillators is our famous one, but there are others as well. So these kind of Swarmalator models were introduced in a number of diverse real-world systems that both sink and swarm, such as vinegar eels, magnetic domain walls, and Japanese tree frogs. So Swarmalators can be both organismal and non-organismal, as long as they exhibit these two properties. More formally, they are dynamical units with spatial degrees of freedom, as well as internal degrees of freedom, where the dynamics are coupled. And so these are some real world examples. This is a graph that kind of shows kind of the different domains where these swarmalators exist and the number of particles involved. So as we go up on this graph, there are more particles per swarmalator, and then we have different domains: biology, chemistry, physics, and engineering. So in biology, we have a lot of swarmalators, as we might expect. We have Japanese tree frogs, magnetotactic bacteria. So these are these are organisms that exhibit these taxis, or these movement towards some sort of source, in this case magnetic sources. We have migratory cells, cells that move, say in development, they move from one location to another. Embryonic cells as well. So we can use this to model embryogenesis or think about embryogenesis in this way. And then, of course, microswimmers, such as sperm or vinegar eels. And so again, these are very tiny units, or I guess in this case cells, or whatever, that can swim in a fluid, and so they have to sort of stick together in that fluid. And sperm are a great example of that, because they have to have this sort of both synchronization and the swarming ability. Chemistry, then, we have chemical pumps and chemical micromotors. In physics, we have magnetic domain walls, or like electromagnetic colloids. So these are like Janus particles, which we talked about earlier in the meeting, and Quincke rulers. And then in engineering, we have robot swarms. So these are rovers or drones. And so I know a lot of people are doing drone engineering nowadays, and they're trying to figure out different ways to build, collective behaviors into these collectives of drones. So you have a bunch of drones that go out, and they might do something like a search and rescue mission, or they might do something like, an attack pattern, and they have to have this sort of both syncing and swarming ability. So this is, it's sort of this diverse set of systems that's kind of being brought together under this topic. And so if we look quickly at the two-dimensional swarmalator model, we have, this is a model in 2 dimensions. So in its generic form, we have these equations. We have the spatial dynamics combining pairwise. So we have the spatial dynamics characterized as a set of pairwise interactions, which produce swarming and aggregation. So we have a bunch of agents in space. They have pairwise interactions. Their pairwise interactions have to be coordinated to get sync, but they also have to be coordinated in space to get swarming. So we know a lot from different types of collective behavior models that they can't be too far apart, they can't be too close together, or sometimes they need to be close together. They send these signals and they know the location of their neighbors. They do this in a pairwise fashion, and then that propagates across the swarm or the flock. And so we can actually use these pairwise interactions in a number of ways to get these kind of behaviors. The novelty is the novelty of the attraction is modified by a phase term F. So this term up here, F. Thus the aggregation becomes phase dependent. So in other words, you have this sort of novelty aspect that gets put into this equation as well. Likewise, the phase dynamics contain a sync term G sub sync, which is here. And again, we have these subtractive relationships modified by a spatial term, so the synchronization becomes position dependent. So it's this difference between these two, I and J. And so you have these pairwise interactions that we can look at. In short, the swarmalators model the interaction between self-synchronization and self-assembly in space. So we have this 2-dimensional model, and we can extend it to the 3-dimensional case as well. The 2-dimensional case is the easiest way to look at it, and we'll see in a minute what those 2-dimensional cases look like. We can expand those to 3. There are 2 parameters, J and K. J controls the strength of the phase space attraction repulsion, so there's this attraction repulsion dynamic. And then K describes the phase coupling strength. So you have attraction, repulsion, and then you have a phase coupling. The above can be considered as a blending of the aggregation model introduced from biological swarming. So in these equations, we have J and K. They represent different things. And then we have this blending of the aggregation model. We go to reference 23. That's this by Chad Topaz and Andrew Bernoff. Non-local aggregation models, a primer of swarm equilibrium. This is in SciAm review. That might be a nice paper to review. So in terms of the phenomenology, this is what the Swarmalator states look like. So we have a static sync state. So if we look at 2 dimensions, we have the static synchronization state. We have the static asynchronization state. So these things are all synchronized. These things are asynchronized, but they're both static. Then the third example is static phase wave. So you have this ring where we have these different phases. And we have this standing wave that kind of moves around the toroid. Then we have the splintered phase wave. So we have this phase wave around the toroid, but they're split into groups, so they're independent phases and they're discontinuous. And then we have the active phase wave where we have this moving, it's no longer static, it's moving around. So we have the static cases, we have the splintered case, which is discontinuous, and then we have the active case, which is continuous. So there's this rotational aspect around a central point. So you can think of fish milling, where fish mill around a central point as an active phase wave. So in an active phase wave, Swarmalators run in a space phase vortex with half running clockwise and the remaining half running counterclockwise. So there's actually 2 phases of motion within this phase wave. So we can think of this in terms of order parameters, and the way they do these order parameter plots is we have K versus the degree of order. So we have this, basically these 2 different types, these 3 regimes. We have the static phase wave, actually 4 regimes. Okay, and so we have 4 regimes here. We have the static phase wave, the splintered phase wave, the active phase wave, and the static asynchronous phase wave. So we have gamma and T minus K, so we have gamma here, this function, which is active during the active phase wave, and some in the static asynchronous. And then we have this black function that is active in the splintered phase wave, decreases during the active phase wave, and then goes to extinction in the static asynchronous. So all of these represent these different states that I just showed you, but these are sort of the order parameters for this. So there are several unresolved puzzles and open questions related to Swarmalators. We have a number of different phase transitions that we want to characterize. The first is what they call melting point, which is K sub m of j. What is the value of this parameter at which the static async state melts into the active phase wave state? So we have the static async state and the active phase wave state. We have this boundary. And the idea is, where do we find the line between those 2 things, or where's the transition? And it's not a sharp phase transition, so that's why they're asking this question. As you can see from this graph, it's kind of just melts into another state. So this is one question that we have. Where do we get these boundaries? Sometimes we get sharp phase transitions and it's very obvious. Other times we get situations like this where it's not quite obvious where that point is. And so this is what we're trying to find. The other is a splitting point for K sub Sj, and that's what is the splitting point at which the active phase wave spills into the splintered phase wave. So again, this is the same question, but only for this side, where again, you don't have you have a sharp phase transition here, but not here. And so it's hard to know exactly what that signature is. And we have rainbow order parameters. And so the question here is, can you derive an expression from the supercritical branch of S for the fixed J and active phase wave and splintered phase wave states? And then N sub C, what determines the number of clusters formed in the splintered phase wave? So in this case, we have maybe clusters like we saw up in the number of states here. We have the splintered phase wave. How many clusters do you have? How many discontinuous discrete states do you have here? So we can actually model this in 1 dimension as well. So we have these 1-dimensional Swarmalator models. This is where the Swarmalators are depicted as dots on this plane. And so we have this one-dimensional model. So we actually have, we have the asynchronous case where there's no one-dimensional pattern. We have the phase wave state where these patterns are kind of oriented diagonally. That's the one-dimensional aspect. We have the mixed, where we have these sort of discontinuous patches along this orientation. And then the synchronized, where they're discontinuous, but they're very tightly organized. So those are what we have here for this one-dimensional Swarmalator model. The Swarmalators are depicted as dots in this plane, and then we have to pick out the one-dimensional aspect. So this simple Swarmalator model is where we can restrict spatial motion to a one-dimensional ring, and according to these equations, and we get these different states. And so then that gives us sort of a door into modeling this with different types of models. So we can actually use the one-dimensional Swarmalator model, and we can actually get some information out of this. But we can also use a Kuramoto model, which models each individual organism or individual agent as an oscillator. And those oscillators have to synchronize. And so this is a Kuramoto model and the one-dimensional Swarmalator model, and it just depends on what you're trying to do. You can look at phase coupling strength of the Kuramoto model. You can use the one-dimensional Swarmalator model, and that has its own advantages. So the other thing I'd like to talk about is the complexity explorables model of Swarmalators. This is Swarmalators, oscillators that sync and swarm, patterns that emerge when collective motion and synchronization entangle. So this is where we have this entanglement of collective motion and synchronization. So this is a demo. This is actually from this paper by O'Keefe, Hong, and Strogatz, oscillators that sync and swarm. This is from Nature Communications, and we'll see this in a minute. It may capture effects observed in biological systems, such as chemotactic microorganisms or bacterial biofilms. And so these are some YouTube videos here of Swarmalators being realized as groups of little robots and flocks of drones. So we can put these into practice. So this is a simulation here that shows a Swarmalator. We have the synchronization strength here, which we can play with. And we have Lycotrex life strength, which is this homophily. So we can set that maybe low or high. And we have these different, I don't know what these are. These are visualizations, I guess. Makes me dizzy, fractions, rainbow ring, dancing circus. Let's try solar convection. And then these are some advanced settings. So why don't we just do this and see what happens, play with it a little bit. So this is the first example. This is synchronization strength very high. The homophily is about average, so you see that they're starting to sort in this ring, or in this sort of spherical thing. It's not a ring really yet. You can see the different states. They're sort of pulsing between states here. This is the solar convection visualization. If we do makes me dizzy, and I'm just curious, this is where they kind of migrate in and out of the central cluster. So it's interesting how they're kind of, you're able to see different types of patterns here where different states are sort of migrating around and providing different shapes. What about fractions? What does that look like? Yeah, now it's starting to form this annulus. And there we go. We're getting this annulus, which is actually what we saw in the demos where we have these different, now we're starting to get individual clusters here, as you can see. So if we go back up to this example here, it's like a splintered phase wave. And that's where it's converging. So that's synchronization strength of average, like a homophily of high. Let's do this. We'll start this over. We'll reset it. And then we'll do a synchronization strength very low, homophily of average. We'll do fractions. And now it's this, again, this sphere. The states are sorting. You can see patterns in there. It's kind of interesting. And so now it's kind of sitting there. It's just, you know, and then let's see if we do like solar convection again. Let's go back to that. Okay, now we're just getting a more rapid sort of sorting, and that ring is moving around a little bit more. See, there's not as much variation in terms of the shape as there was before, so this is interesting. Synchronization strength again is very high. The homophilia is about average, so now we're favoring sort of synchronization of behavior over sorting into different states. So naturally we would see maybe something more like this static sync state. All right, so that's the simulation, and then this is how it works. Here we have n equals 500 Swarmalators, so we can't vary the number of Swarmalators in the simulation. It just runs from there. We can vary the other things. The state of the Swarmalator n is defined by 3 variables: the internal phase and 2 positional variables, x and y, because we're in this 2-dimensional regime. The phase is depicted by a color of a continuous rainbow color wheel. So, you know, that's where we're getting this pulsing of color. Initially, the Swarmalator's phase variable is random. All of them place randomly in the plane, and all are at rest. So they all start at sort of a resting state, an initial condition. And then, you know, you apply the equations of motion, and, you know, they get the simulation. So the movements are subject, or the Swarmalators, are, in terms of movement, are subject to 2 opposing forces. We have short-range repulsion, which is where, again, we have these pairwise relationships between Swarmalators. We get this difference, and the difference determines whether we see repulsion or whether we see attraction. So, you know, this is where this homophily comes into play. We can set this so that it favors maybe attraction, it favors repulsion. But we do have these 2 opposing forces, and so they get balanced out in different ways. So the first is short-range repulsion, where 2 Swarmalators come too close. If they come too close, a repulsive force dominates and pushes them apart. So this allows them to not bump into one another, but it also results in them moving away, having a tendency to move away. This force is negligible when Swarmalators are far apart, obviously, because there's no reason to push them apart. Additionally, any 2 Swarmalators experience a force that pulls them to get towards one another. This magnitude of the attractive force does not decrease with distance, which is why it's a long-range attractive force. The clue is that this attractive force between 2 Swarmalators, say n and m, depend on their phase difference, or this difference between m and n. When like attracts like, parameter j is positive. This is this homophily here. If it's positive in this direction, a similarity in phase enhances the attractive force. When this parameter is negative, homophily is down here, the Swarmalators are more attracted to others of opposite phase. So these colors, you get these, either you get these clusters of like type, or you get these sort of regions of variation in type. And then, of course, we have our synchronization, where there's the Swarmalator phases advance at a constant phase velocity or frequency, like an internal clock. So we can, you know, map this in biological systems to like clocks, like, you know, cell division clocks, or other types of different types of oscillating mechanisms in biology. Additionally, a Swarmalator's phase also changes as a function of the phases of other Swarmalators. So when the synchronization parameter k is positive, the phase difference of m and n between 2 Swarmalators, n and m, this decreases as they tend to synchronize. So this phase difference, as they synchronize, the phase difference decreases. So basically you get, instead of having an anti-phase sine wave, you get an in-phase sine wave. And when they become desynchronized, the opposite is true. You go from a synchronized sine wave to an anti-synchronized sine wave. This type of phase coupling coupled oscillation is also explored in the explorables Ride My Kuromoto cycle and Janus bunch. And so this is the Kuromoto oscillator, Ride My Kuromoto cycle. This one here, looks interesting. It kind of rotates around like a cycle or a motorcycle, and it's running around. It's you can set it so that it synchronizes. A set of oscillators, and then this Janus bunch. And we talked about Janus particles earlier in the meeting again. This is where the oscillator has two faces, each corresponding to a distinct internal phase variable. So again, this is where we have these, these two phases, and they're kind of moving around in this sphere, bouncing around, changing size and color. So it's lots of fun. And so yeah, you can run through this complexity explorable. It's very fun to do. Now let's get to the papers. So there's this first paper here that is from O'Keefe, Hong, and Strogatz. This is oscillators that sink and swim. So this is sort of the seed paper for this. So this is one of the first papers that really kind of synthesized a lot of this area in terms of a review. So Stephen Strogatz has been talking about these kind of oscillators for many years, and so, but this idea of a swarmalator is really a newer idea. So synchronization occurs in many natural and technological systems, from cardiac pacemaker cells to coupled lasers. And we can see this, and this is sort of the power of the heart, I guess, as it were, is that the cardiac pacemakers are synchronized, but they also behave collectively. And then, of course, with coupled lasers, it's the same thing. In the synchronized state, the individual cells or lasers coordinate the timing of their oscillations, but they do not move through space. So there's this temporal aspect, but not the spatial aspect. A complementary form of self-organization occurs among swarming insects, flocking birds, or schooling fish. Now the individuals move through space, but without conspicuously altering their internal states. So this is where they're moving around, but they're all kind of in the same state nominally. They're just trying to coordinate their movements. And so this is where the very classic models of Boyd's, where you're just simply modeling the interactions between individual agents. You're not worried about their internal state or any other type of state. Here we explore systems in which both synchronization and swarming occur together. So we have these models like cardiac pacemakers modeled with flocking birds. And so we combine those two. We take the interactions, but we also take the internal state. Specifically, we consider oscillations where phase dynamics and spatial dynamics are coupled. We call these swarmalators to highlight their dual character. So this is the kind of the linkage to the Janus particles. You have these two different characters of the swarm. A case study of generalized Kuromoto model predicts 5 collective states as possible long-term modes of organization. So that's what we were talking about in the Wikipedia article, that these 5 different modes or long-term modes of organization that we were converging to in the complexity explorables case. These states may be observable in groups of sperm, Japanese tree frogs, colloidal suspensions of magnetic particles, and other biological and physical systems in which self-assembly and synchronization interact. And we saw that graph of the different types of systems earlier. The second paper, and this is the last thing I'll talk about today, this is Dynamics of Swarmalators, a Pedagogical Review. This was in 2022 in the European Physics Letters journal. Yes, this is in the EPL. This is actually, so this is also introducing swarmalators. Swarmalators have emerged as a new paradigm for dynamical collective behavior of multi-agent systems due to the interplay of synchronization and swarming that they inherently incorporate. So this is getting into this multi-agent systems modeling. And this is an interesting area, not just from the standpoint of collective behavior and self-organization, but also from the standpoint of coordinating agents in something like reinforcement learning. And so we've been doing in the orthogonal lab, we've been talking recently about multi-agent reinforcement learning and how you can model that in different ways using different policies. So this is a nice sort of rejoinder to that work and thinking about this in different ways and how this can be implemented and how it can be explored. We have this dynamical collective behavior of multi-agent systems. There's this interplay of synchronization and swarming, and their dynamics have been explored with different coupling topologies. So this is what we were talking about earlier with these coupling topologies, where we have these phases that we can extract, these global phases, interaction functions, external forcing, noise, competitive interaction, and from other important viewpoints. So there are a lot of things that you can explore. They talk about in the paper these interaction functions, external forcing. We didn't actually look at the perturbation button on the complexity explorable. There's a perturbation button in the simulation here, this perturb, which I guess just means you kind of move it around, you kind of push them in different directions. So you disrupt, if this were to converge to one of these annuli, you could break apart that annulus and reconverge the simulation, which is interesting because it can give you different results. So we have these different ways you can explore. Once you have this basic model, then you can explore the dynamics quite a bit, and there are a lot of interesting things going on here. There's also noise, of course. We can introduce some sort of stochastic mechanism or set of stochastic mechanisms, and sometimes noise can help synchronize the systems as well. So it's not just always disruptive. And then we can have competitive interactions where we actually have like strategies, conscious strategies, or some sort of cognition of some type. So just strategy making and things like that. So it's just really interesting stuff that you can do here. Here we take a systematic approach and review the collective dynamics of Swarmalators analytically and numerically. Long-term states of position aggregation and phase synchronization are revealed in this perspective with some future problems. So this really goes through a lot of stuff here. Goes through some models of Swarmalators as the bidirectional interplay between the spatial and phase dynamics of the Swarmalators, trying to sort of model interparticle collisions and trying to take those into account. We have this KJ parameter region. We're looking at it a little bit different way than the Wikipedia article. This is where we have J versus K, and we have these different stable states here: active phase wave, splintered phase wave, static sync, and static async, and their relative position in this phase space. So we can say for low K and high negative, or for high negative K and low positive J, we have the static async phase. We have the static sync phase for all positive K below 0.6. The static phase wave is kind of at higher J and maybe low positive K. Splintered phase wave is low negative K and high positive J. And active phase wave is this other region here, this leftover region in the positive J. So this is, we can break these states down into this phase space, and we can, think about this in terms of homophily. So for positive J, like attracts like, which obviously if we want to swarm, we have to have some sort of homophily at some level. We want to sometimes have repulsion, but we don't want to do this in the absence of attraction. So this, you can think of the phase space in this way, and we can kind of figure out where these states are. And so here we can look at the transitions of states as a function of force amplitude. So this is F as force amplitude, where we can actually calculate sort of how these transitions happen according to a single parameter. So they come to the conclusion that theoretical analyses have revealed that under certain assumptions, such systems possess some conserved quantities and finite time collision avoidance can be assured. The long-term states of these systems have been analyzed numerically and theoretically under global interaction, addition of external forcing, local interaction, and competitiveness and phase coupling, with both static and time-varying coupling topologies. And so more studies like oscillatory behavior through repulsive short-range interaction, active phase wave with attractive phase coupling, dynamics under stochastic coupling in memory, and mean field limit have enriched this field, which we mention here and we're unable to discuss in our main text. And then finally, we talk about some of these things that we need to explore. Swarmalators with endowed orientation can reveal new collective states where their soaring properties include both aggregation and orientation. There are these three exponents, alpha, beta, and gamma, that determine the asymptotic states. The role of these states and how they come to be is not fully studied yet, which demands more attention. So as you tune these different parameters, they produce these stable states, and we don't really know why that is yet. A new but impactful direction of research would be to consider non-pairwise interactions among the units, where higher order interactions takes over, and that's citation 53. So we have 53 down here. This is from the Journal of Royal Society interface from 2022. I don't have the title, but that's the citation. Maji et al. So this is an interesting thing because, we think in terms of these pairwise interactions, but at the global level, we also have to think about modeling that. And sometimes it's separate from these local interactions. And so the stability properties of active states of the 2D Swarmalator model remain one of the aspects in which demands sincere attention. Looking at the difficulty of analyzing the 2D model, one can ask, can we model a solvable system for the Swarmalators? So maybe it's not a solvable system. Maybe it's something we can look at in silico in a simulation and derive insights from, but we may not be able to actually analyze all of the interactions. We can use 1D models to get there, but that's not the 2D model, and it's certainly not the 3D model, and that's where we want to get. So that's all I have to talk about with respect to Swarmalators. I hope you found that interesting, and I hope you learned something.